And then the sequence that you can use
I can send you a picture with you. Send something to me. Okay, this week. Okay. Okay, yeah, what's up? Yeah, okay. Good.
the sequence that's not working. Give me a second, Magnus. Give me a second.
While these guys sort out the technical problems here, I want to wish all of you a warm welcome. Christian Brechot is a very busy man. He will be here in time to inaugurate the Blitz science section, and that will be a real challenge. We will have 5-minute talks followed by 5-minute questions. I will be a very hard chairman, as I always am. This morning we had a steering committee meeting for the International Network for Data Analysis here in Pasteur. And as you know, Institut Pasteur is not one institute in Paris. It's 32 institutes around the world in 25 countries, and this is an expanding network. And we discussed many collaborative projects this morning, both in research, in education, and in outreach. And we are very convinced this is a network for bioinformaticians and data analysts. And we are all dedicated and convinced that data analysis and bioinformatics will be in the forefront of deepening and enhancing the collaboration in this network, because the network constitutes something that is unique. It's a unique global asset with biomedical knowledge, with data that is absolutely unique in its breadth. And this is something that no research institute or organization around the world can match. So if we really use this and go together, we will be able to provide something unique to the society, but also, I think, show how science will be done in the future already now. So this is about globally sharing data and expertise, really pulling together pieces and create a puzzle or a puzzle that we will lay together, and I think that will be something that no one else can match if we decide to do it. There are several projects that are ongoing, and we have a website that is soon coming up that will be hosted in Montevideo, where we will show all of these projects and efforts that we are doing jointly. So I think that will be exciting. Are we more or less on?
No.
Nothing works.
No.
Okay.
Do you know how to work? Rachel, how did you make it? You just work directly?
Should be working.
It should be working, yeah.
It should work. It just no. The button in the middle. Yeah.
Yeah, we're using more than the 10 minutes that we had given to us by Christian, but that's okay. This works now? Yeah. This works now. That's already a wonderful improvement. So I can just give you some updates while everyone else is running around. So we have several things that we've been discussing this morning. Among them, we have a possible partnership with Macrogen, that is a sequence provider, where we are looking. This will be a multi-step process. We are not targeting just one company, but we are targeting several companies. Macrogen is one of them, and I think that it is a wonderful opportunity to create closer links that could provide added value in many of the projects where we need to have a service provider. Because if we have a close relationship where we can actually go together, even on the research level with the service provider, that will give much better quality to what we do. We also have been discussing a partnership with Fiocruz and University of São Paulo, and this is moving forward quite rapidly. So probably we will have a tripartite partnership signed, maybe already this summer, between Fiocruz, that is a network that is as big as the Institut Pasteur network, and the University of São Paulo, which is one of the biggest universities in the world. We are also moving forward on establishing PhD student exchange in the network within bioinformatics and data analysis. And this is just a couple of small things that we are currently doing. So I think that really, are we ready to start? Okay, wonderful. Things are more or less working. So we have an interesting agenda. We have a couple of talks in the beginning that are a little bit longer talks, and then we have really short blitz sessions after the coffee break that will give you all a smorgasbord of what is being done when it comes to data analysis and bioinformatics within the network. So again, and now more formally, all of you, warm welcome to this session. And I invite our first speaker, Artemis Hatzigeorgiou from Greece. She will talk on regulation of regulators, characterizing the promoters of microRNA and their targets. Warm welcome, Artemis. Okay.
So, thank you for giving me the opportunity to be here. It's really a great new action with all this podcasting. So, I'm coming from Hellenic Pasteur, Greece, and this is 100 years old. So this is one of the first buildings. Here is actually my lab, and we have also new buildings there. And let's speak about microRNAs. So what are these microRNAs? They are 22 nucleotides long RNA sequences, and they came to really change what we know about molecular biology. They came to change the dogma, where we have DNA making RNA. Let's see where I have it. DNA making RNA, making proteins. And now what they do is they stop the production of protein even if we have the messenger RNA. So where are they located? They are also processed from the DNA with long transcripts, and then coming out of they do in hairpins, they form hairpins very fast in the nucleus. they're cutting out from DROSA, the hairpin is coming out on the nucleus in the cytoplasm, then with more proteins, DISA is cutting the loop, and they're having, at the end, we have the 21 nucleotides with the RISC and the Argo complex, and they go on the messenger RNA. Oh, that's great. So the pointer came. Now I have to hit. That's it. So, and they bind on the messenger RNA where it has or it cleave it or it repress the translation. Now, how is it doing it really? We have many options or many ideas of how it's done. I will say a few of them. Maybe, so messenger RNA is making more a cyclist, and they stop the ribosomes from scanning the messenger RNA, or they let the ribosomes, they abolish the ribosomes, they stop the production of the protein, or they produce the cleavage. Now, what is the, okay, why we have the microRNAs? What is their function? So imagine now 150 or 300 or 400 proteins are on, and we want them at one point to have them shut down. How we are going that at once? So a microRNA is processed, it binds to the messenger RNA, stop the production of protein, and slowly the production of messenger RNA stopped also. So also, if we have a protein expressed in a high abundance at a stage of our life, and we want it later to have it in a lower level, what we can do is also process this microRNA and then just lower the expression of this microRNA. Saying that, if their function is disrupted, they can be involved in many diseases, as we see metabolic disorders, causes of obesity, and stem cell proliferation and development. They're important. Let's stay in viral infection. So also the virus have microRNAs, and what happens is this is the way how they interact with the host, having their microRNAs bind on proteins of the host. And in this way, change the regulation of protein coding genes in the host. So microRNAs came up, like in 2002, we just learned that we have many microRNAs. How many at the time? 100, 110. And so we see how the development is until today. So you see here the miR-base entry. This is the basic microRNA database. How we learn how many we have. Right now we say we have like 1,500 or 2,000 in human. Though it's still a question how many of them are they important and functional. And with the time also the people who are working on them also and publish on them is increasing. So let's go again on their biogenesis. They are located or intragenic, and they have their own transcript transcribed with pol2, polyadenylated transcript very often, and different transcription factors in front of them, or they are intergenic, no, actually, that's how I ran. So, or they are in the introns of protein coding genes, and they're getting transcribed mostly with the messenger RNA of the gene, or they have their own promoters and they're getting transcribed with their own transcript. So why is it a challenge to find them? Because, as we said at the beginning, if they have their own promoter, already in the nucleus, DROSA is coming and cutting this hairpin structure out. So what happens is the rest of this transcript, that can be even 40,000 or 100,000 nucleotides long, get degraded. We don't need it anymore. So if we normally get with CADES the full transcripts, we get transcript for messenger RNA that stay much longer in order to produce protein, but we don't get this full transcript that disappear very fast. So how can we predict the promoters of this transcript? Let's go back to promoter prediction. What did we have at the beginning for protein coding genes? We had at the very beginning weight matrices of transcription factors and how frequently they are in front of the transcription start sites of protein coding genes. Then the information about coding region came and splice junction, and then CADES data came in order to really sequence the start of the messenger RNA. Now, going to the microRNA genes, we had at the beginning to go back to our first methods, looking for weight matrices of transcription factors, and then the new area, this great area of ChIP-seq data came and epigenetic data. So now we could use the ChIP-seq data from Pol2 occupancy, and we could use the data from histone modification, which are really the small, we can say, lashes that we see here that change dependent of the activation of a gene at that point. Saying about ChIP-seq data, let's say in few words what is done, for example, for Pol2. If we have a Pol2 on the DNA, we can cut them with immunoparticipation, then get with an antibody the Pol2. At the end, we want actually only the places where the Pol2 binds, sequence them, we have them, map them of the genome. We have these places where the Pol2 has bind on the DNA. How does it really look on real data? So here we see how Pol2 looks. Here, H3K4 in a long non-coding RNA that we have here. The drawback is that this can be really broad places in order to identify the transcription start site. What we need to know in order to see what transcription factors are regulating this gene, protein coding or non-protein coding. So how do we validate a microRNA transcription start site prediction? As we said, the problem is that we don't get these transcripts very easily. So it doesn't matter what programs we have, and there were few programs out. The main point is to know how good we are in what we're doing. So what we did was to engineer a new mice in collaboration with Regeneron Pharmaceutical and Aris Economides there, where we have a depletion of the DROSA. If we don't have DROSA at all, the mice is lethal embryonic lethality here. So having these conditional mice, it was possible then to get cells where we see what happens if the DROSA don't catch the microRNA. And what we really could see was the longer transcript. So here we see on the H19 link RNA what happens on the DROSA plus mice. We see a little of coverage, but if we do the DROSA minus, we see that the transcript is staying much longer, and we can get the information about the whole transcript. And here is the occupancy of Pol2, and here is the occupancy of the epigenetic information, the H3K4. Now, having finally few data where we know where the transcripts are, we looked on a lot of published data out there, deep sequencing data. And what occurs to us was that if we have really deep sequencing, we can see something of these long transcripts. So here we see what happens with 27 million reads and 19 million reads for DROSA minus and DROSA plus. But if we go to 180 million reads, then we can get a big part of this full transcript. So this could be something, a point to go on without to have to do conditional mice again. And now if we have this information from really deep sequence RNA data, still we have a question, which is the putative TIS? Because real data are not so nice as our graphics here, our schema. So we need more information to decide where is the real start. And what we can use there is add the information from H3K pheromone, the epigenetic information, the information from Pol2, and the transcription factors footprints. Only a very short word about what this is. This is made with DNA sequencing, and the idea is that as we have here the DNA wrapped along the long nucleosomes, the transcription factors can bind on the places that are open. With DNAs, we can get longer places around the DNA, nucleosomes, and shorter, which actually are the transcription factor footprints. And this can help us as additional information where transcription factor binds in front of a transcription start site. Now, how the algorithm works. On the first step, we get all the RNA-seq, map on the genome, then in species, make pieces together where we see that we have a quite bigger occupancy and have the putative transcription start sites. Then we go and see what is the distribution of the information we have from the epigenetic data and the POL2 around the transcription start sites from protein coding genes, where we know where the start is. And we can define places where we say this is a positive and this is a negative example, and again positive and negative example. Having a set of positive and negative examples, we can finally go to that where my expertise from longer time is machine learning. So if we don't really understand what happens, let the machine learn it. It's nothing different as we do with our brain. We just learn by example, positive and negative. And that is what we're doing. We're using a support vector machine, and it learns, depending on the information it has from the deep sequencing data, where is the transcription start site and where not. We're putting this all together this is the overview, the first step, the RNA-seq. Then we have 3 vector machines, support vector machines, say for each state, if it could be a transcription start site. And finally, we get the first from the microRNA, starting from the microRNA that goes above the threshold, that can have all the information to be a transcription start site. Now, how good are we on this? Looking on the protein coding genes, we are getting very good. Okay, this looks so nice. So what is really, is a prediction or the test set perfect? What happens here is that our first on the protein coding genes looks all nicer, and our negative set is nearly so big as our positive set. But the reality is not like that. Normally, we look for one positive in hundreds and thousands of negatives, and that makes the problem much more difficult. When we really apply that on the real data that we have from the DROSA knockout, we are getting pretty good. And here, that is what we get, getting a sensitivity. So how many we get? We get 44 out of the 47, and we get 100% actually precision in what we say, what is correct. And in actually here, this is 3 other programs that are available right now, or were available also when we published, about transcription start sites of microRNA promoters. And if you see here, I mean, here again, let's go to the sensitivity, which is how many are the correct predictions to the total, so how many of the transcription start sites we really get. And this is the precision where out of the predictions, how many are correct. You see here that you have few programs have much more predictions because they predict more sites than one, and we had a way to count it. So we said everything that is around 1,000 nucleotides from the transcription start site is correct. Now, let's to close this section of the talk to say that when we look the data on the human data, what we could see is that we had microRNAs that are not annotated as a transcript, that are read through with known genes, others that are exonic, other intronic in the introns, and others that are at the end of annotated microRNAs. We have others that are intronic normally and intergenic, and very interesting, we found also a group of microRNAs that are located, that we could identify the cross-seq data and are located on the opposite side of the start of a protein coding gene. Now the question is, of course, how many of them are really functional? Interesting, 1 or 2 of them, they are also conserved in mice, other of them not. And if we see here how the evolution is dependent on the group we have, then we see that the one that are divergent on divergent sides, they are newer, they appeared much newer than the others on the genome. And this is a recent publication, came out in December 2014 from my group and the 3 people from the Regeneron group. Now what else we have in the group? Actually, we are working on microRNAs in many years, since 2001 in fact. So we have a microRNA target prediction program. We have a database of experimental supported targets on protein coding genes. And we have a database from long non-coding RNAs, targets of microRNAs on long non-coding RNAs. We have the annotation of microRNAs on the genome, miRPath, which did an analysis of targets of microRNA overrepresented in pathways of KEG, and in our newer version that is now under review on Go ontology also. Then we have association of microRNAs to disease. Here we have also on miRPath, we have also map to SNPs. And we have a tool for microRNA target gene enrichment analysis, where once you see that you have a group of genes differentially expressed between two stages, you may want to see if this is caused because these genes are changed expression and mainly target from a group of few microRNAs that are responsible for this change. And a microRNA naming history, because microRNAs change the name very frequently. In the last years, they disappear, they come, and extended connectivity to online databases. Out of this, these are the primary data resources, and this is the meta-analysis group. Few words now, I will go very fast, about target prediction and what is the problem. Actually, why these microRNAs are there? They are in order to drive all this whole risk complex exact on a place on the messenger RNA is where the microRNA bind. Actually, we have a RNA binding with a messenger RNA. So what is the challenge on microRNA target prediction? To find exact displays, and what you use is thermodynamic binding, and then energy, and then we see if it's accessible, how conserved the site is, and many other things. We are doing this actually, as I said, many years, and we found recently that also coding sequences, particularly if the UTR is short, could host targets of microRNAs. I can say that from a work with Zyssimos Mourelatos, we have the patent on finding the targets of microRNAs, and this is a good story why computational people together with experimentalists should work together. This was the time I was at UPenn as a professor, and a new professor was also Zyssimos Mourelatos. He asked me to do a prediction, he validated, we had it. It took 10 years to get the patent, but we got it. Now, this is how the web page for microRNA target prediction looks. So this is how the target looks, how many targets we have, how conserved. You can go to the Santa Cruz browser and have the view. There's information about what else is about microRNAs or genes in the literature, accessible on microRNA.gr, microRNA TCDS. This is the TAR-based database of the experimental validated targets. I have to say that the new database that just appeared like a few months ago has now 500,000 experimental validated targets. And this explosion of targets came through a lot of clip data based on ARGO that came up in the last year. So if microRNA binds to ARGO and we make chipsec of ARGO, then we can get the microRNA binding site. And this is data that we've analyzed a lot. Having TAR-based the biggest database of experimentally validated targets, Ensemble asked us to integrate our data. So right now the target prediction for microRNAs on Ensemble is based on our annotation. Another tool is the pathway analysis. If you get hundreds of targets for microRNA, you want to see these targets, is they overrepresented in a pathway, and this is what this tool is doing. So these are the particular targets of one or a group of microRNAs in a pathway. Several other tools are available here. I will just say that we have Taverna plugins of our tools available, and seeing here that Galaxy is available here, it will be possible to change this plugin to Galaxy and have it incorporated in a workflow where actually starting the analysis from RNA-seq, you see what is differentially expressed, and then you can see overrepresentation of microRNAs, immediately go to target prediction, pathway analysis, SNPs. This is an example of how such lines would go with microRNA gene names, sequence data, the sequence information, micro-T, see the targets, involvement of microRNA, see the results, go to extra, miRPath, and continue. I will not say so much into that. this is another example how someone can start with a target of microRNA and using the pathway analysis to filter it, or to go from a gene that someone is interested in, see which microRNA targeted, and again, what is the enrichment of the targets of the microRNA that target this gene. Just to say that we had this summer a training school on non-coding RNAs in Pasteur, Greece, and actually we had more requests that we could host, so we can do it again. It was very interesting. How many people access the microRNA GR, our web tools? We move from one place to the other, so I have two analyses. Actually, this is per month. We have now something like more than 12,000 people visiting and using our site per month. And here is an older distribution of users from many places around the world. And with that, I'm coming to the most important part of the lab, the people behind them. So this is Giorgos Georgakilas. He's finishing his PhD in 1 or 2 months. He is the first author of the transcription start site prediction program on microRNAs, an expert on NGS, regulation microRNAs, and he is looking for a postdoc somewhere in Europe. Yannis Vlachos is the postdoc of the group, also worked. He's doing all the pathway analysis, tarbase, and regulation. Maria Paraskevopoulou is a PhD student. She will get her PhD in something like half a year. Did all the link-based analysis, tarbase analysis, and not a lot of RNA-seq. Dimitra Karagouni started half a year ago with SNPs on cancer. And Konstantinos Liakos, it's a master student, he just joined. So thank you very much for your time. Oh, I thought there will be one more time. So.
Okay, thank you very much. Questions? Comments? I have a question. So, I mean, contemplating how microRNAs work and the pipelines you already have in place, do you see clinical applications? I mean, that could be...
Yes, I mean, why we're looking on the promoters of microRNAs, why we're looking on microRNA targets? Because if we look for disease, of rare disease, and we see the differences, different polymorphisms, most of them are located in no-coding regions, and we want to understand which of these 200,000 is the one that caused the disease. So if they are on the transcription factor of a microRNA transcript, the only way to find them is know the promoters. Or if they are on the targets of a microRNA, you have to know where the target is, and see if this one SNP, which happens, can abolish the targeting of the microRNA and on the messenger RNA. So in this way, they are very important.
Okay, let's thank the speaker again. Our next speaker is David Hart from Institut Pasteur Lille in France. A pipeline to quickly and automatically characterize microbial genomes from high-throughput sequencing data. So please.
Okay, thank you very much. I would like first to thank Magnus first for this wonderful network and for giving me the opportunity to talk today. So I'm David Hart. I'm working in a lab which is called Transcriptomic and Applied Genomic at Institut Pasteur Lille. We are linked to a center which is called Center for Infection and Immunity of Lille. And today I'm going to talk about the development of a tool which is a pipeline to characterize automatically microbial genome starting from high-throughput sequencing data. Most of the work I'm going to present is the work of Ségolène Caboche, who is a PhD in my lab. And I'm not myself a bioinformatician, so if you have a specific question about mathematics and bioinformatics beyond what I'm going to say, please ask questions, and Ségolène will be pleased to answer these questions. So I think I don't have to tell you that. You all know that we are living something revolutionary in terms of sequencing with high-throughput sequencing, and we are able now to generate a tremendous amount of data very quickly and for a cost which has nothing to do with what was this cost a few years ago. And even with a small sequencer, such as ion torrent PGM, you can generate a sequence of microbial or to sequence or resequence microbial genome. So there's plenty of new application emerging from that. Whole genome sequencing or resequencing, it's what I'm going to talk about today. Target sequencing, RNA sequencing, chip sequencing, etc., etc. So when we started to work with this kind of data, we realized that the difficulty was not really to get the sequence and the data, but more to make sense of that, of course, and to be able to compare what we've got when we sequence a new strain or a new bacteria, for example, and to be able to compare this data to what was already in the data bank, and to find what the new sequence genome was sharing with other annotated genome in the data bank, and what was specific for this sequence in terms of gene content, plasmid content, mutations, insertion, deletion, etc. So we decided that we could try to develop a pipeline to do this work, to have something which is efficient, fast, and automatic to characterize microbial data, starting just from the HTS data. And this kind of pipeline can have, of course, some interest in different fields, in epidemiology, of course, and let's say clinical microbiology, to characterize new variants. And there's several groups over the world who are trying to use this HTS as a characterization of new strains, such as GMI. You probably know about it, Global Microbial Identifier. You can have some interest as well in molecular biology research when we generate or we select new mutants and we want to know what's going on at the level of the genome. And of course in phylogeny as well, when we want to characterize and compare some collection of strains. So you probably know that for all these approaches, the main part of the difficult task is to go from the raw data, which is the FASTQ data here, where you have the reads and the quality assigned to the read, to the mapping of the reads. So it means that the algorithm the mapper has to localize correctly the reads on the reference genome. And this seem to be an easy problem, but actually it's not, because we are working with short read, as you know, and the reference genome is very big compared to this short read, even when we work with microbial genome. And in addition of that, we are in a case where we are not with an exact matching position, because there's the sequence errors, the sequencing errors, and there's a mutation, and that is a point we are interested by. So there's the sequence errors and mutation, and there's some repeats region in the genome which create some noise for the mapping as well. So one thing we realized is that there was a lot of mappers when we started this work, but the characterization and the assessment of these mappers was not really available, or if there was some assessment, there was mainly focused on Illumina reads and mainly in the context of human genetics. So it's not exactly the same question when you are dealing with microbial problems and microbial genomes. And so we decided to see how to choose correctly an appropriate mapper for the question we wanted to raise. And so there was plenty of parameters to test. And Ségolène in my lab developed a complete protocol to assess, to make a comparison of different mappers using both simulated and real data sets, so simulated reads and real reads, and using a new definition of what we can call a correctly mapped read. She tested 14 different mappers, and you can find the whole study in this paper. So what we are doing as a protocol, we start with simulated and real data sets, the reference genome, and we apply or we test different criteria. We looked at the computational resources and time required to do the mapping. We look at the robustness of the mapper, meaning the sensitivity and specificity of the mapper. We looked as well at the behavior towards the repeats in the genome and to the way it deals with mutation position. So today I'm going to focus mainly on this part, the robustness, which is the most part. I'm going just to tell you a few words about the criteria. So as I said, we used real data. We obtained this data on the ION community, which is a website for people who are using and sharing information about PGM ion torrent sequencing. And we used 3 sets with different mean lengths of reads because we wanted to see if the read length has an impact on the quality of the mapping. And we used as well some simulated data. To do that, we developed a new tool which is called the QSIM for customized read simulator. And this QSIM is able to generate synthetic reads mimicking all the reads generated by the different kind of sequencer. So you can generate any kind of reads. It starts from a genome you give it to him, and is going to generate some reads with some insertion, deletion, and substitution at the rate you decide to get. Then you have a small additional tools which is QSIM enroll to evaluate the generated set of data, and you can find these tools on these websites if you want. So using QSIM, we generated this simulated data, where, as you can see, we have changed the level, the rate of insertion, deletion, and substitution. So we finished with 9 different set of data with a different total error rate and different combination of kind of mutation, kind of error, let's say. And so with this simulated read, we ran the different mappers and we looked at the position we obtained for the different reads, and we decided to change a little bit the definition of the correctly mapped read, because so far in the literature, people were using only the start position as a criteria. So we decided to use start position, but also end position and number of insertion, deletions, and substitution in the read to define a good, a correctly mapped read. Then we calculated the precision and recall for all the mapping results. And we calculated a F measure, which is a harmonic mean between precision and recall. So here you have the results of the behavior of the different 14 mappers according to the rate of mutation. As you can see, most of them hopefully behave quite correctly because they are able to position correctly the whole set, or almost the whole set of reads on the reference mapper, on the reference genome, sorry. But as soon as you introduce a little bit of error, substitution, some of the mappers are starting to give less good results. And if you add insertion, deletion in top of substitution, you see that some of them are behaving and giving very wrong results as mapping. hopefully a few of them were able to stand the pressure, let's say, and be able to give a good mapping of most of the set of the data, even with a very high level of mutations or error, let's say. We tested as well this kind of mapping with real data to see if our simulated data were strong. And you see that we have the same kind of behavior as soon as, so we used a program which is called Rabema, developed in 2011, and which gives an interval of position for each read, and then you can calculate the percentage of retrieved intervals in function of the error rate. And you see that, like with simulated data, we have some mappers which are not able to map correctly the set of data, and some of them are able to keep a good result even with a high level of errors. And we observe the same ranking with simulated and real data, so which means that the reads we generate are close to the reality. So we tested different aspects as well. As I said, we looked at the memory required to run a given set of data, and you see that from one mapper to another, we have a very, very different requirements. We looked at the time needed to run, and here as well, it goes from a few minutes to several hours of run, so different need in time for a run. And we looked at behavior towards the repeat, so we generated a artificial genome with 5 different repeated regions, and we looked how the mapper were able to position the reads on this repeated region, and you see that only a few can find 5 out of 5 position on the genome. So, as a conclusion on these mappers, we don't have the perfect mapper for everything, but the conclusion is more that some mappers are better for some criteria than some others. For example, some are very quick, but not as the best or the robust, most robust one, and some others are quite robust, but needs a bit more time, of course, to run. So depending on what you need, the best way is to use a combination of mappers, and actually it's what we did for the pipeline we wanted to develop. We used different mappers for the different steps. I'm going to go through the step of this pipeline. So this pipeline is working like this. It starts with FASTQ data, so reads. We give him as well the whole reference genome as a FASTA and GFF data, and plasmid sequence as well as FASTA and GFF. Then there's 4 different steps. The first step is just to check the annotation and format to be sure we can work with the different file we have in the data bank. Then there's a first important step where we are going to find let's say the phylogeny, which means what are the closest reference compared in terms of genome compared to the sequence we are analyzing. It gives you as well a list of present and absent CDS in your genome and a list of mutation. I'm going to come back on the different steps. The second step is doing exactly the same, but for the plasmid, if there's some plasmid detected. And finally, there's the third step, which is analyzing the reads which have not been mapped at the previous stage to see if there's some additional gene or part of genome, such as horizontal transfer gene or insertion. Then there's some other module which are running, such as a de novo module, which is going to to deal with unmapped reads. There's a comparison model when you are working with different genome and analyzing different genome. And there's an alert module which is creating a kind of antibiogram. I'm going to come back on that. So the first step is doing is finding the closest reference or references, and is generating the list of CDS present and the list of mutations. So what it's doing is to do first a mapping using a quick, rapid mapper using SNAP to compare the whole set of reads against the whole set of genome sequences. So it has to be quick to have a rapid answer. Then once we have found the closest genome, we do a more robust mapping using SHRM2 to obtain the list of mutation compared to the closest genome and the list of covered and uncovered CDS. And you can have a graphic output like this, which gives you the gene of the closest genome which are present or partially present or absent compared to the sequence you are analyzing. The same kind of analysis is done with the plasmid, if there's some plasmid in the data. And there's a third step where we are doing what we call an iterative mapping, meaning that after the two first steps, the unmapped read, if there's still some unmapped read, the reads which have been mapped on the closest genome and on the plasmids are mapped on the second closest genome, then on the third closest genome. And at the end of that, if there's still some unmapped reads, we are using them in what we call the de novo module. So the de novo module is just an assembly, a de novo assembly of the left unmapped reads, and then we blast the obtained counting on a data bank. We use Patrick CDS to be able to complete the picture of the genome we are dealing with. There's as well some a comparative module just to be able to make some comparison in terms of list of genes and list of mutation between the different genome. And we developed as well, it's still in progress, we developed an alert module, which is a kind of e-antibiogram, and which gives you actually a comparison to the presence of genes compared to data in a drug bank, which contain drugs and their target genes, and ARDB bank, which is the antibiotic resistance gene database. So depending on what it's described in these two data banks, and depending on the presence or absence of target genes and resistant genes, we can try to predict what would be the phenotype of the strains we are dealing with compared to against the resistance or sensitivity to drug or antibiotics. So just to finish, we used this pipeline on a real case. In 2011, there was a short outbreak of enterohemorrhagic in Lille, in the north of France, and we were able to get some clinical isolates from this outbreak, 3 clinical isolates, and we sequenced them using ion torrent technology, and we run it on the pipeline. So here you have an idea of the time needed to get the full analysis of the 3 different genomes. So it's very rapid, and what we saw is that we got of course the same bacteria than the one which was detected by classical approach. So it was a 0157H-, but we had a much more precise idea because we knew exactly what was the profile of the plasmid, and there was actually a different profile in the 3 different isolates. so different plasmid contents, and as well, one of the isolates was the STX2 gene, so shigatoxin 2 gene was absent on this isolate compared to the two others. So we have something which is much more precise, of course, than the classical approach, and in a time which is very interesting. So, as a conclusion, I show you that we did a full protocol of comparison of mappers, and we get some interesting results of the different mappers. We finished saying that a combination of mappers is very often a good solution when we are dealing with high-throughput sequencing data. We develop a tool for fast characterization of microbial genome, and this pipeline is not yet available. It will be very soon, and so you can check for what's going out for this kind of approach. I would like to finish by thanking people of my lab, so mainly Ségolène Caboche, who did this work, and Christophe Audebert, who is really implicated in this project. And I thank as well Michel Simonet and René Courcol from the hospital who give us access to the isolate. And I thank you for your attention.
Okay, thanks a lot, David. Questions, comments?
So it's a very nice question. I don't know anything in the field, but so I was surprised that of we isolate for the same outbreak. Is it common? And it suggests that if you try to apply this kind of clinical application operations, you will have to sequence everything.
Yeah, well, exactly. We don't really know if it's common, actually, because this kind of full sequencing is not usually done on isolates, and people with just the characterization of the bacteria, we usually have enough to deal with clinical problem, and we don't try to go further. But this kind of approach show us that probably it's a bit more complicated than what we think, and when we look carefully to the whole genome contents, we can see some differences. We could have some importance for the clinical treatment.
Okay, so I will take the opportunity to also pose a question. So, I mean, I didn't really get exactly how you evaluate, I mean, the quality scores in your different, when you evaluated the different mappers. Mm-hmm. But more importantly, that I didn't get is, I mean, there is a rapid technological development, so we get longer and longer reads with better and better quality. So, did you look at some
longer?
Yeah.
Third generation,
yes.
Or, yeah. Well, it's something we are planning to do. We are in the, for example, in the Oxford Nanopore map program to get some sequence with this technology, with so very longer reads, but probably more mutation or more errors and different kind of errors. So this kind of protocols we develop is really easy to apply to this new technology, and it's something we are planning to do very soon. Yes.
Thank you. You did a wonderful job in mapping comparisons.
Thank you.
And indeed, one of the strengths of mapping is that you can attach qualities to SNPs. Mm-hmm. But one of the problems with SNP approaches for epidemiology is that is long-term sustainability. So, for example, how will you compare the next outbreak with the previous one if your pipeline chooses a different reference for SNP code? Then you will have different sets of SNPs and are not compatible.
Okay, so I didn't say it, but you always have the possibility to, in addition to the reference the pipeline is going to find, you always have the possibility to impose one of your reference in addition to the one it's going to find. So if you have one reference you want to be part of the analysis, you can add this reference in the list, and it's going to take it as a part of the set of analysis. And so from one time to the next one, you can keep the same reference. Yeah, it's true. You are completely right. And there's as well the database which evolved, and so every time we have different set of genome to work with, so it's going to change from one analysis to the next one. But we have the possibility to keep one reference genome if you want to. Okay, thank you.
Okay, let's thank the speaker again. So I think that was a wonderful example. I mean, everyone that works with mapping knows that you use different mappers, and you do this ad hoc and by hand. So I think this is a wonderful initiative, and hopefully we will link to it very soon from the INDA webpage as well, so that people in the network can start using these tools that we develop in different sites. So it's my pleasure to introduce our next speaker. This is Guilherme Oliveira from Belo Horizonte in Fiocruz in Brazil, and he will talk on exploring helminth genomes for drug and vaccine development. So please, Guilherme.
Thanks so much, Magnus, for the chance to be here and talk about my favorite project. And I wonder, I'm very envious of our last speaker. Can you imagine how cool it is if people ask you about your work and say, just look it up hot in PubMed. Okay, now to a serious tone. Do I change it? So the reason I chose this topic is because it's a nice story from the early work we did on schistosomes when we wanted to learn about its genome. That was in the 90s. And the whole aim there, with the support of the World Health Organization, was that we really did not have a complete set of tools to control this disease. So getting the genome sequence was something that would open opportunities to explore new ways of combating schistosomiasis. And the good part of the story is that since then we've moved a long way to producing what's now a helminth reference genome, and also to make a dent into the development of these new control tools. So I'll spend one slide to talk about my home institution if you are not aware of it. Fiocruz is a foundation. We are part of the Ministry of Health in Brazil. We are distributed throughout Brazil into a large network that contains production facilities such as pharmaceutical industry and biopharmaceutical industry. We have a presence in Africa, and we have 16 different basic research units distributed throughout Brazil, and I am part of one of these units, and we have colleagues here who are part of additional units. So it's a rich network, and I think that what is being discussed here today fits really well with what is being proposed by my colleagues at Pasteur, and I hope we can make this a very active joint activity. Well, now on to schistosomiasis. This is a huge disease globally. We have over 200 million people infected and some 600 million at risk of infection. It's second only to malaria as far as its impact on human health. And it's responsible directly or indirectly to over 200,000 deaths per year. So it has a huge impact in global human health. The current status of control of the disease is basically treatment. The problem is that we have one single drug to do mass chemotherapy. It is a good drug. It kills about 90% of the parasites in people infected, but people get readily reinfected. It doesn't kill the larval forms of the disease. And if we've learned anything from our colleagues in the veterinary field, who work on helminths is that having one drug is no comfort because resistance will appear. We have no vaccine to combat schistosomiasis, although there's lots of active work going on there. And as I'll point to you later, it's a very interesting organism because we've been looking at ways of exploring the different approaches that the parasite has to adapt to its environment. So it has to live in fresh water in two different life cycle stages, the larval infective form and also the eggs. It lives in a snail and also lives in a vertebrate host, and it has to migrate through the host. It has to communicate with the host and receive signals from the host that trigger different maturation steps or physiological adaptations. So it's a very interesting model organism as well. So resistance can appear, and we have shown that in the laboratory, where you can see that if you pressure the parasite with increasing amounts of praziquantel, we do get parasites that resist to 3 times the lethal dose that's used normally. And that's done in a way that you restrict the genetic diversity of this parasite. And people have seen similar things in lab settings and also in field settings. We never get everyone treated in the field. So parasites that somehow are less sensitive to the drug are there. The question is whether, with increasing drug pressure, as we see being happening in Africa, is if they will a similar situation that's developed for malaria will happen in schistosomes. So the first thing we needed to do is to produce a genome for this parasite. So we've produced, with a large international consortium, the first version of the genome in 2009 and a revised version in 2012. And also for Schistosoma hematobium, another one of medical importance in 2012 as well. So here's a comparison of the 3 main species of medical importance. They look pretty much similar in terms of size, gene numbers, etc. This is a figure that shows the relative evolution from version 1 to now version 5 of the genome. Here's how the scaffolds were in version 1 and how, in general, we were able to close many gaps and increase the size of the scaffolds and close gaps with the original version of the genome that was done using Sanger sequencing, now with short reads. So we've gone quite a long way into developing, this is the best reference we have at the moment. And also to describe how these genes are transcribed along the life cycle of the parasite, because obviously if you want, for instance, to create drug targets, you want to target proteins that are expressed at the inside the vertebrate host. So we know now the genes are present, they're expressed in many of the different life cycle stages, some of the core genes that are always expressed and some that are specific to some of the life cycle stages. So this is fairly well mapped, although there's still more work to do. One of the issues as we produce large data sets, and I only showed some of it, is to integrate this data set so that people can make use of it, and not only hardcore bioinformaticians. So what we did with the genome information was to create an environment, a database environment, where this information could be uploaded and then searched using a web-based tool. So pretty much the quick version of this, we use the Genomics Unified Schema database architecture in Oracle. So pretty much we decide what we want to upload. There's a number of plugins that put the data in the respective tables. We run automated analysis. The results are parsed and uploaded again. And then we can mine the database either directly through using SQL, or we pre-compute queries that are available through the web interface. I won't go into too much detail on how the database works, but this is just its front end on version 3. We're now, soon version 4 will become online. So pretty much the cool thing about the database is that as it is relational, you can combine queries very easily and create some very complex queries on the fly. So this has been well used by the community, and it's available free to anyone who wants to investigate these genomes. Well, one of the things that worried us from the beginning was genome annotation. As with many other species, about at least half of the genome was a total blank. We had no idea what the proteins were doing. So what we did was to run genome-wide phylogenetic analysis in order to derive annotation from other genomes and upload it into schistosomes. So I won't, again, go into too much detail. It's been published. But we created what it's called a phylome-DB, and through it we could derive then information from 17 other full genomes of model organisms and upload them into schistosomes. And in addition to it, we could also investigate gene families that were either expanded or contracted in schistosomes, and that gave us some interesting ideas on phenomena that are that this parasite evolved and adapted to deal with this specific life cycle needs, such as some protease families that are used to penetrate the skin of the host. We also use that information, that early information, to derive a metabolic map of the parasite using the pathologic tools. And we could not only query these pathways and overlay expression data onto it to understand what's happening on the metabolism, but also investigate things that were happening that were specific to this parasite. So this is just a general metabolic map that you can drill in and find genes and compounds. And I'll exemplify with a couple of ways we've interrogated this database. So one of the first things we queried was, well, what is it that the parasite produces that is unique to it? So that those enzymes that are responsible for those compounds could be targets of interest. So we have a list of products and enzymes that are produced exclusively by the parasite. And one example here is this compound 2-phenylethanol. Interestingly, it's what's responsible for floral scent in roses. But I'll tell you, schistosomes don't smell anything like roses. But it's also used in insects for communication, and it's one drug that's been in clinical trial for cancer treatment. And over here, it may give us some insight into how these parasites communicate with one another. But then these are enzymes that could possibly be targeted, and by being unique to the parasite, would be interesting targets. Another way was to look for choke points in these metabolic pathways. So a choke point would be something like this, where one compound is only produced by one enzyme reaction or consumed by only one enzyme reaction. So by drugging one or the other, you could make the compound accumulate or not exist in the parasite. So we've looked at the entire metabolic pathway, and we came up with a number of pathways that were choke points. And we picked up a few of drugs that were directed to these choke points in order to test whether this would work. And we picked up a number that worked, at least some of them to the extent that praziquantel worked in the mouse model. Obviously, no one's going to treat schistosomiasis with methotrexate, but it just demonstrates the point. So there's a list there that people can go and test. It's available online. Now, more recently, we teamed up with an international consortium funded by the FP7 program to develop drugs aiming at one specific group of enzymes. These are histone-modifying enzymes. So these are modifying enzymes that modify histones by writing things into it with acetylation, methylation, phosphorylation, and others, or they delete these modifications or read these modifications, and that results in some action on the genome. So these are global methods of regulating genome expression. So the first thing we had to do was to identify these enzymes, and the brief story is that we aligned, created hidden Markov models, and conducted phylogenetic analysis to annotate the enzymes. And we came up with a list of enzymes for all 3 species, and we picked up some that were our first case study. To demonstrate that drugging these enzymes would harm the parasites, this is with a non-drug TSA, and you can see here that by in a dose response manner we can kill parasites with TSA, and this targets SM deacetylase 8. And so we started from there and then investigated other targets to see if they were interesting. And we did that by first demonstrating that the presence of these enzymes was important, and we did that by using RNAi. Schistosomes is still not amenable to knock in and knock out, so RNAi was the best we can do. And here's an example of some of these targets, and you can see that when we do RNAi on HDAC8, our favorite target for now, and some other histone-modifying enzymes, we can either decrease the number of adult worms that we recover from infected. So we treat the parasites in vitro and then put them into mice and see how much we recover. So this diminishes the number of recovered parasites or the number of eggs produced. And actually the eggs are the cause of the pathology and not the adult parasites itself. Well, using HDAC8 as an example, I'll show you briefly the pipeline that we developed. This also is not done everything in my lab. This is a large joint effort. So one of the pathways that we use is by using X-ray information to derive drugs. So we clone the enzyme, we produce the recombinant protein, process and purify the protein, and then crystallize this protein. And from the crystal information, and we crystallize it pure or with inhibitors, people can then go in and modify these compounds, produce the compounds, and we can test them in vitro and in vivo. So we can, some of the things we've done with the virtual structures was to screen libraries, and one of them is the enamine library with 1.6 million compounds. If you're interested, this is what was used to do the in vitro screening, and we derived compounds from this assay. The other approach was to do high-throughput screening, again with the recombinant protein that has to be active. An assay was developed, and that assay was used to do high-throughput screening on a library of about 300,000 compounds. And so we have an assay, and then we screen these compounds, and we come up with structures that can be then synthesized and modified to screen the parasite. So I'll give you just this one slide. I failed to put the publication here, but this has been published. This is the parasite HDAC8, and this is the human HDAC8. So they're very similar enzymes, but if you look at this phenylalanine here on the parasite, it gives a different binding pocket compared to the human enzyme. So this small but significant feature was used to derive compounds that would then be have higher affinity to the schistosomal protein in comparison to the human protein. So if you look here, this is IC50, so you want the bar to be low for parasites and high for humans. So you see here that with SAHA and TSA not shown here, we had the compound was active, was inhibited the parasite enzyme, but was more it hit the human enzyme better than the parasite one. But with the first generation compounds, we could almost reverse that entirely, where the affinity was much higher to the parasite enzyme in comparison to the human enzyme. And now we are in the third generation of compounds, and this has really gone in different ways. So we have now good compounds that we are comfortable in saying that these are lead compounds that we can now push further into perhaps clinical trials. Well, the SETRAN project finished about a year ago, and we started with a new round of funding from the European Commission on the A-Paradise project. And what this is allowing us to do is to expand the same types of pipelines now to other disease models such as leishmania, malaria, and Chagas disease. So we're developing compounds that target all these diseases. I won't go into much detail here, but in parallel, we've been exploring kinases. So if you remember, the parasite has to interact with its environment and adapt to its environment on a constant basis. So to do that, it has to capture external signals and translate those signals into some physiological adaptation. And that necessarily goes through kinase proteins cascade. So we did the same to identify the complete set of kinases in a similar manner that we used for histone modifying enzymes, and we have now the complete kinome of the parasite. These are very well-behaved enzymes, as we can easily classify them. And again, we use RNAi to validate some of these enzymes as targets. So you can see here with junk and ERK, we can decrease the numbers of worms recovered from infected animals, and with again here with decreased numbers of eggs produced by parasites exposed to RNAi. This slide just shows how variable RNAi is in schistosomes. So this is something we have to deal with because the system is an outbred population. These are adult worms that they vary wildly in their behavior. So just go with the ones that perform well, such as these ones and the ones that were tested. We also, when using ERK and junk RNAi, you have a hard time seeing it, but the fact is that we can disrupt the outer membrane of the parasite to a large extent and also disrupt the reproductive system, especially of the females. So that's why you see cessation of egg production almost complete here. So these are the lines that we're going along on developing compounds for kinases as well. And recently we received a grant to do high-throughput screening of these kinases in collaboration with the University of Nottingham. Other things that are on the way, we have funding from the NIH to do a 5-year follow-up on the fields. So we go to the field, we treat people, we collect material, parasite material, and we want to see how a 5-year treatment scheme will impact the genetic diversity of this parasite, and perhaps we can map what's related to the appearance of drug resistance. But this project is not only aimed at investigating parasite genetics and drug resistance. What we're doing here is producing a protein array with 1,000 different proteins produced in vitro to screen the entire population and get a perception of what's the immune response to each of these proteins, so that we can derive then candidates for vaccines and also diagnostics. Diagnostics is a huge issue, especially in countries like Brazil, where the endemic areas is not diminishing, but the level of infection is diminishing. So we are pretty much working on the edge of the sensitivity of the diagnostic assays available, which is egg counts in fecal material. So these are being produced as we speak, and we are currently also doing deep exome sequencing to understand, as I mentioned, population genetics, but also antigenic variation on these 1,000 proteins, but also on the targets that we are developing drugs to. With the FlatTB consortium, this is a consortium from Argentina, Uruguay, Brazil, and Australia. We've developed genomes for other species and are pushing this just behind schistosomes to develop new compounds. And I mentioned the kinase work. So with that, I'll be wrapping up, and I would like to thank people in the lab. As I told you, all of this is a large effort and highly collaborative. So the TMRC was funded by the NIH and would allow us to look into the field for 5 years. The NIH also funded other small collaborations to develop parasite genomics. The European Union for funding the drug development projects for using histone modifying enzymes as targets. The INCT Brazilian Network for Tropical Diseases funded some of the field work that we carried out. The FLAT Genomics Consortium funded by Argentina, Brazil, and Uruguay. And another grant from the NIH that allowed us to develop the schistosoma genome database. With that, I thank you. And thanks again, Magnus, for the chance to be here.
Thanks a lot, Guilherme. So questions, comments?
So what do we know about the microRNA?
Okay. I actually have one paper on that. They are there. We find many microRNAs that are common to other species, but we find many new microRNAs that have not been classified yet. More recently, some groups in China especially did, when we published that paper, it was only Sanger sequencing, but now we have some next-gen sequencing on that. So no one knows exactly what they are doing, but they are there, and we have lots of new ones.
Any other comments or questions? Okay, we'll thank the speaker again. Thank you, Guilherme.
Thank you.
So we go for the last talk of this first part, and this is Cheikh Loucoubar from Dakar. So he's you are a new G4 group leader in Dakar, and you will talk to us here about multi-way gene-gene interaction analysis for family data. So please, Cheikh, welcome.
Hello, everybody. Thank you for giving me the opportunity to talk here. I will present you a method to search for gene-gene interaction in family data. Let's first define what is epistasis. For example, that means interaction of genes. Betson first define epistasis as a phenomenon of masking effects. It's when the allele E in one locus prevents the allele in another locus from manifesting its effects. And later, Fisher present epistasis as a deviation from additivity in the effects of allele at different loci with respect to their contribution to a quantitative trait. In a statistical point of view, we can define epistasis as a statistical interaction. That means a departure from a specific linear model that describes the relationship between the covariables. Here, covariables will be the different allele at different loci. Until now, we have a lot of results published after GWAS studies, and the thing is, few sets of interacting variants, that mean epistasis, that explains the disease risk was identified in these studies. And we know that a great part of epistasis should be implicated in the infectious disease trait, which are in general complex traits, multifactorial traits. To investigate for epistasis in large data sets, we need some specific techniques like multi-marker approaches and methods like machine learning, including logic regression, multidimensionality, multifactor dimensionality reduction methods, and random forests have been used to solve this problem. But there are some main issues in searching for epistasis in large data sets. Beyond the issue due to computational power, we can cite the multiple testing problem. The number of combination of alleles increase with the number of markers involved in the epistasis that we are looking for. And some solution is to limit the search of interaction in some candidate genes or candidate pathways, or to limit the analysis in just the statistically significant genes that have significant marginal p-values. But doing this, we can miss epistasis, especially for markers that do not satisfy the pre-specified criteria. We also have the problem of sparsity. genotype combination. One solution adopted by the MDR method, that's the method that I cited before, the multi-dimensional, the multi-factor dimensionality reduction, is that it collapses genotype classes, and this can lead to a loss of precision. So why we are interested in searching epistasis? It's because we face a malaria dataset in the context of the malaria genomic epidemiology network. And the data that we have in Senegal is come from a longitudinal familial data. So I plot in the y-axis, you can see it's the ID of the person, and it's just a picture of the survey on some individuals in 1 year, for example. And the line means that the person is exposed, he's present in the site, and each dot is a visit. If it's black, he is negative for malaria. Blue, he is positive but asymptomatic, and red, he is positive and sick. So this is like, so we have longitudinal data, and also people that we are following are genetically related. So they are also, we have trios in the sample. So that's why we have within and between individual correlation in the data that we have to take into account when we do analysis. We also have some population admixture issues because we have different ethnic groups in the villages that we are following. For example, we have Serer, Mandeng, Wolof, Fulanis, and some other ethnic groups. One solution to analyze these data is to use the TDT approach, the transmission disequilibrium test. We know that it's a method that sometimes lack of power, but it is not affected by population admixture because we are testing transmission of alleles from a parent to an offspring, referring to the parent's genotypes. But generally, this test is designed for 1 p-allelic markers. And for the traits that we have, we expect to have the joint effect of several markers. So that's why we want to develop an analysis method that fits the best the design of the data that we have collected. So let me just make a quick reminder on what is the transmission disequilibrium test. The design is we have an independent parent affective offspring trios, and we have we have 1 B allelic marker to test. So we have allele A1 and AB, A1 and A2. And if we define like P12 is the probability that a parent having genotype at the marker A1, A2 transmit to his offspring the allele A1, and N12 the sample frequency of these events. The null hypothesis is the Mendelian law, that means P12 equal P21. The same, we have equal probability in transmitting one allele instead of the other. And there is a known statistic to do that, which is the McNamara test, which is a chi-square of DF1. There is a likelihood version of this method. So we can write the model into this likelihood, which is the probability of transmitting 1 instead of 2 at the power, the number of times that events occurs, time, the opposite. That means parents transmitting the allele 2 instead of 1 at the power, the number of times that event occurs. And then we have the log likelihood. We can derive easily the log likelihood of this. And under the null hypothesis, we see that P12 equal P21 equal 1/2, so it's minus log of 2, etc. And the test statistic is the log likelihood ratio based test, which is also a chi-square one, similar to the McNamara test that we presented before. So what we have here in our data, so we have N independent parent-affected offspring trios, but we have K multi-allelic markers to test simultaneously. In general, we have SNPs, so they are B-allelic markers, but we developed this method to be able to test like haplotype that can be considered as one multi-allelic markers or also microsatellites. So given that you have the markers 1, 2, 3, L1, L2, L2 until LK, with each marker having a number of alleles, like small L1, small L2 for each one until LK. So we can now, for example, the maximum number of combination set of allele that a parent can transmit to an offspring. So screening the population, we can have at maximum the number of different sets will be the product of the else, number of allele at the first locus times the number of allele at the second, etc. until the end. But in general, we will not observe all the possible combination of alleles. For example, given that a parent have this genotype configuration, at locus 1, the genotype is A prime, locus 2, B prime, and until locus K, V prime. And we know that A and A prime will take values in 1 until L1, etc. So if we look at a couple of parent and an offspring, we will observe that one set of allele, for example, S equal A, B until V is transmitted while the other one is not transmitted. For example, if we are studying 3 B allelic loci, we will have 8 possible triplet that are 111, 112, etc. And we have to screen the data set and count the number of times that we have, for example, for a parent who is homozygous at the 2 first loci and heterozygote at the 3rd locus, some of them will transmit the triplet 112 instead of 111, and some other parents also will transmit 111 instead of 112. And the idea is to test if this is 50-50 or not. For people knowing what is the TDT test, they know that there are the issue of when the 3 member of the trio are all heterozygous, we cannot know exactly which of the parent have transmitted, for example, the mutant allele. But it will not affect the transmission count, so the test will lead to the same thing. But here in a multilocus setting, it can have an impact. We can consider this scenario. For example, we consider that for this offspring, at locus 2, the father gives a small b, or another scenario, well, it's between Mac and PC, move a little bit. Another scenario, we consider that the father gives the allele b. So we can have 2 different consideration that will impact the table that I showed before. In the same way here, we can define, so for that problem also we have to, we can solve this problem. It is integrated in the programs that we have developed by considering all possible configuration as equiprobable, and then also correct the weight, the transmission count for this specific trios. So if we define like MSS prime, the frequency of informative parents with respect to S and S prime, that means parents that have not exactly the same, that are not homozygous at all loci, those parents will not contribute to the test. And NSS prime will be the number of parents that have transmitted the set of allele S instead of S prime, and the probability which is associated with these events is PSS prime. So we can screen all couple of S and S prime by considering all A less than, so all A and A prime in this domain 1 until L1, and considering also A less than A prime. So the likelihood, the log likelihood of the transmission model I can say the joint transmission model is given by this formula. And under the null hypothesis, we have this. And we have the test statistic also, which is developed as log likelihood based ratio statistic of tests. And the thing here is we have to define now what is the function, what is the probability of transmitting the set of allele S instead of S prime. It will be distributed as a chi-square, but the degrees of freedom also will be a function of the number of allele depending on the alternative hypothesis that we will consider. We can see in the next slide that we can have several different alternative hypotheses when we are testing many markers together. So we will present the asymptomatic test statistic, which is based on the formula that we showed. We will also present empirical test statistic, which is based on simulation, by simulating the transmission of allele from parent to offspring at each locus by using the Mendel's law of 50-50. And we see that they are very close. So these test statistics, the theoretical statistic of tests that are asymptotically distributed as a chi-square is not like an aberration. It's very close to the reality. So when we consider the k loci, we can decide to test only the highest locus, ignoring the information from the other loci. And for that, we have to define the probability of transmitting at that locus the allele J instead of allele G prime. The probability will be the transmission intensity of the allele J divided by the sum of the transmission intensity. And the test statistic will be a chi-square with Li, the number of alleles at that locus, minus 1. And this will be equivalent to a previous test developed by Sham and Curtis in 1995, the extended TDT. It's the TDT for one marker multi-allelic. But we can consider, in our context, we can test among the k loci a subset of q loci, and q can be equal to 3 until k. And if we consider the set of q loci, we can test them, their joint effect in an interactive manner or in an additive manner. So it depends. If we consider that the set of q alleles are independent and they contribute to the trait in additively, the probability of transmitting a set of allele A, B, V will be a function of the product of their marginal risk of transmission. So it is defined by the first one. And in the second case, where we are, when we consider that the joint effect of the set of allele is in an epistatic manner, there is the risk, the intensity of transmission of the set of allele is read on the table of the transmission count. And here it is just the number of alternative hypotheses that we have when we consider a set of k loci. So when we consider a set of k loci, and if we want to test the k loci simultaneously, we have a number of alternatives by considering all possible configuration on this k loci, and it increased very quickly. Like for 5 markers, we have more than 50 possible models that we can build on 5 markers. So we will consider corrected p-value by using the FDR correction method. So to evaluate the method, we did simulation studies. So we consider for the simulation 3 biallelic loci, and we test them on different configurations. So we make 5 etiology. So by etiology, we refer to the model that we introduce in the trait, in the simulated datasets. So we simulate dataset by introducing effect in the traits And we introduce 5 different effects. Like sometimes we just put the effect in one of the loci, and sometimes we put the effect in 2 of them, sometimes in 3 of them, and sometimes additively, and sometimes in an epistasis manner. And we do all this 1,000 times for each configuration. And also we cross this for 3 different minor allele frequency, 3 different sample size, and 3 different effect size. But the effect size, we put it in the single. We put different effect size in each single SNP, and additively it will be derived. But in the epistasis scenario, we simulate a deviation from the additive effect size. And this is the results of the power study. So we compare the method, the MTD method, with the MDR PDT, which is the most close method to analyze the type of data that we have in the literature for now. The MDR is, this method MDR PDT is a combination of 2 methods, the multifactor dimensionality reduction and the pedigree disequilibrium test. And we look at this. This is the results at effect size 1, at effect size 2, and at effect size 3. And what we observe is, I have some problem on, okay, with the colors, okay. But in general, we have, for the single locus scenario, the MTDT is always more powerful than the MDR PDT, and also have lower error rates, family-wise error rate. We have lower false positive also. And for the multi-locus scenario, when we consider a trait which is underlined by an additive joint effect, the MTDT is also always more powerful and have lower family-wise error rates. And the in an epistasis context, the MDR PDT is more powerful, but it is accompanied by a high rate of false discovery. And if we make a summary on all the simulation studies, we can find concerning the power, the MTDT has a mean power of 88%, while MDR PDT is at 57%. And for family-wise error rate also, the MTDT is at 3%, while MDR PDT is 7%. So in summary, the MTDT is more performant than the MDR PDT to analyze the same data. So in a family-based setting, the MTDT is well adapted for hypothesis-based testing, because the method can distinguish in a context of a joint effect, it can distinguish between additive effect and epistasis, while the MDR PDT will just detect joint effect without saying if it is an epistasis effect or an additive effect. And it is also this method is well defined for genome-wide screening. So as results on the malaria data that I presented to you before, so we have repeated measurements on all individuals. So we just run in a first step a mixed model to pick up the residuals, and the residuals we split it in the epsilon and in an individual effects also, the random effect, and these individual effects will capture all the effects that is due to the individuals. So we take out the effect of the variables, the covariate, all of this, and just use the gamma I in blue, which is the individual effect as a phenotype. So this is the distribution of these individual effects. And we have, like, for people having a negative value, they have a negative personal contribution to the disease risk. So they are defined as resistant individuals, and the individual having a positive personal contribution to the risk are defined as susceptible individuals. we can see that in a context of a quantitative trade also, the MTDT method is designed to consider the level of the quantitative measure. So we can weight the transmission count of one parent to an offspring by its phenotypic value. That is good for people that are borderline. For people that are very close to zero, for example, they will contribute a little bit in the statistic of tests, because people at the extreme, we are more sure that they are resistant to malaria. So this is the describe of the sample size that we have analyzed. So we have 276 individuals comprised by 135 persons who are the parent status, and 147 offspring. And we have some people being, so we have the data on 2 generations. So some people have the status of offspring and also the status of parent. And we have 11 independent families, 73 nuclear families, etc. Voilà. So this is the first screening results. So we first consider 2 mutations known to be implicated in malaria: the HBS mutation, which is responsible for sickle cell on chromosome 11, and also 1 SNP on CR1. One SNP on CR1, which is in the chromosome 1, that is also implicated in severity of malaria. And we screen we screen SNPs on the chromosome 17 on the gene SLC4A1, which is responsible in blood disorders. And we also take plus minus 50 kilobases around the gene to capture some SNPs that are close to the gene. So when we did that, we pick up the most significant SNP in this gene, in this SLC4A1 gene, which is the RS207, 41, and 06. And we consider these 3 mutation for a multi-locus model to test the joint transmission of alleles at these different loci. So for 3 loci, we have 11 model possible that are presented here, testing each of them individually, or all the possible couple in an additive manner, and the triplet in additive and in epistasis manner. And here we can see that when we consider, because for the TDT, the number of informative transmission is based on number of heterozygous. So if we consider more SNPs, we increase the number of informative transmission, because for a parent, once you are heterozygous, at least at 1 SNP, you are informative. So this is this will impact the power, so we'll have more power. And here we can see that the three locus epistasis and the 2 locus epistasis have good results. In conclusion, we see that MTDT is able to distinguish between all possible alternative hypotheses involving up to K markers, and have good statistical power across all scenarios that we have tested. And it is accompanied with low family-wise error rate. So we are a little bit protected with false discoveries, false positives. And the increase in the complexity of the models with the number of markers is accompanied by an increase in the number of informative transmission. So in an increase of a sample size, so it will compensate the complexity problem. And for MDR-PDT, it will identify markers involved in the combination with a high statistical power, but also with a high family-wise error rate. So using the MTDT, we detect 3 locus in epistasis, so HBS, the mutation on CR1, which is also just published this year in the Nature paper describing the African Variation Genome Project. They also found the same SNP as one of the most differentiating between Europe and Africa, and also this SNP on SLC4A1. So single locus would have missed the RS, the mutation on CR1, and we detected in using multi-locus approach. So the three epistasis model showed very high power compared to single locus methods with a difference of 2 on the p-value log scale. So the recommendation after these the simulation studies are that in a family-based study, when we have genome-wide data, we can use in the first step the MDR-PDT, and then in a second step specified hypothesis and run MTDT to increase the power of detection. One limitation is that MTDT assumes independent trio, while MDR-PDT is accommodates trio or SIP pairs. So I thank people involved in this malaria project, the people in Institut Pasteur de Dakar, the villagers, the field workers, and also people from IRD in Dakar, the University Paris-Dakar, where I did my PhD in conjunction with the Institut Pasteur à Paris, and the staff of, and also the École des Hautes Études en Santé Publique, who also participate to finance this work. Thank you to my units here when I was in Paris, Anavaj Sakuntabai and his team. Some of them are here, Isabel. I don't know if Rick Paul is here. Oops, sorry. If you have any question.
Yeah, thank you, Cheikh. So, questions, remarks?
Thank you for your presentation. I would have a question about dealing with population genetics. You know that in African countries like ours, you often have to cope with missing data. Because sometimes when you do a follow-up study, you have to face that problem of villagers or people moving around and or not coming back for the follow-up studies. So I do not fully understand how the model, or could your statistical study take into account that missing data?
Unfortunately, we haven't yet included that in the program. So we consider that all parents and offspring have genotype information at all loci, which will limit the things, because we have to drop people or trio for which there are missing genotypes. But some other methods include that, like FBAT, you can do that, but FBAT will work on one locus or multi-locus, but on haplotype, so they are not independent to avoid a lot of combinations. And we in the future we can include, we will work on that to include like inferring genotypes when they are missing. Yeah.
I have a question.
Yes.
Often in these kinds of studies you do a replication, but have you done that?
Not for the moment. We have just, this is the preliminary results that we have, and we are working on a paper to submit these results. But we are more focused in the method than in the biological results. It was just for application, but for sure it will be better to replicate this. But also sometimes design are very specific, so maybe you will not find exactly the same design elsewhere on malaria. The thing is, in general, you have to fit well your data to make valid statistic on your data, and if you can justify that the method is valid, so it's not very bad, but it's better to replicate, sure.
Okay, so if there are no further questions or remarks, we thank the speaker again. Thank you. And I think it's a wonderful example also of where you should precisely use all the biological knowledge or the a priori knowledge that you have to design your model, because it is very hard to get statistical power in these types of studies. So I very much look forward to see what you will accomplish in the future in this. We will very soon go to the coffee break, and I've heard that being a mathematician myself, I've heard that mathematicians are just machines turning coffee into theorems. I think it was actually Erdős that said that. That might be true, and I think that coffee breaks and interactions are extremely important. And we have a challenge in this network because we are spread over many continents, many countries, and it is hard for us to take these coffee breaks together and gather around the coffee machine. But we will have to build these virtual coffee machines in some way so that we really can interact. But we should also, of course, make the most of the possibilities to have these live interaction that we will have now. And the next steering committee meeting will be again in Paris, actually in October, in conjunction with another meeting. And then we have just decided this morning that steering committee meeting for INDA, with possibly also a scientific day, will be in Seoul, in Korea, in the spring of 2016, hosted by Hakim and his team there. So we very much look forward to that. After the break, and we will resume precisely on time, so it will be a little bit longer coffee break. We resume at 4:20, and you will have a wonderful opportunity to, in 7 times 5 minutes, get a sweep over what we are doing in the network. And this will actually cover all 4 continents and 7 different countries. So warmly welcome back 4:20. The coffee you will find at the entrance. So please make the most of the coffee break. My friend Paulo Carvalho from Curitiba in Brazil, and you will talk on SimXL, a powerful and user-friendly tool for peptide cross-linking analysis. Please, Paulo.
All right. Magnus, thank you very much for having me here. It's great to be here in Paris. So I'm from Fiocruz, from Carlos Chagas Institute, Fiocruz, Paraná. So my friend Guilherme already gave a very nice introduction on what is Fiocruz, so I can move on, telling what is this all about. This is probably the most important slide in my talk. So what is this? What are cross-linkers and how are they helpful? What can they do for you? So there are two main applications in what we do. One is the structural characterization of proteins, so helping model the tertiary structures. And the other is for protein-protein interaction. So briefly, what's going on from the bench side, you have a protein who you want to characterize a tertiary structure, or you want to find out interacting partners and where they are interacting. You can add these cross-linkers, chemical cross-linkers that are like molecular spacers, and they will stick to certain amino acids. And you can identify where they stuck to each other in the mass spectrometer. It's a covalent bond. And with these space constraints, you can know exactly where the proteins are interacting, or the spatial constraints will help you get tertiary structure information. This is complementary to state-of-the-art techniques, crystallography, but many proteins are not amendable of obtaining a crystal, or you need lots of quantity, which is pretty much hard. So I'm just going to zoom through the slides. I just learned that if you do not have fancy graphical user interfaces, it's very hard for your software to be adopted. So we put much effort into fancy graphic user interface, where basically the user adds the parameters. And this is the search result. So actually you have a 2D interaction map. So these are, here we are characterizing a 5-protein complex. So here are interlinks showing where you know exactly the information from which amino acid to which amino acid is interacting. You also have a list of, you can also see the results as a list of the mass spectra with the scores and all that, and where in the sequence that these cross-linkers are mapping. One of the nice things of our tool that we just published like 40 days ago, it is the first tool to work with PRIDE. PRIDE is the proteomics identifier, which is the largest international repository for storing proteomics data, developed and maintained by IAMBO EBI, and currently there are more than 2,400 projects hosted and more than 42,000 mass spectral experiments deposited in PRIDE, and we are the first tool to make cross-linking data compatible with PRIDE. So we work together with the PRIDE team to define the specifications on how to store cross-link database, and make it globally available. So this is our first tool. Ours is not the first cross-linking tool, however, it is the first graphical, fully graphical tool. But we compared it with competing tools. And in particular, Crux is the most well-known tool. So in a searching a database where we present decoys, these are the first 50 spectra. Our tool were able only to get confident spectra, while the competing strategy at spectrum number 10, according to ranked according to confidence, it already began identifying decoy identifications, showing that we have much better sensitivity than the widely adopted Crux, and our search speed is orders of magnitude faster. This was in a simple search, but there are searches that can take up to a couple of days. So here would take a couple of, this would take like a week, and this would take like a day. And so these are like 40 days ago. We have this, which is basically describing the algorithm, and this here is one on how to use the software. So these two came out almost simultaneously, like 40 days ago, and we have an avalanche. As soon as we got it out, we have an avalanche of users, and this is statistics from 40 days ago, and it also enabled us to fire up collaborations with institutions around the world already using our software. I'd like to note that this is basically work of Diogo Borges Lima, which is a PhD student I have, together with, he's co-advised by Dr. Fábio César Gozo from Unicamp São Paulo, and we also have some other collaborators from Fiocruz and the Federal University of Rio de Janeiro, but this is the hands behind SIM, Diogo Borges Lima. And that being said, if I manage to, I have a video trying to demonstrate the tool, if I manage to get you going here. I have some, I don't know if it, but it's just. So the user is setting up the parameters in our tool, and just basic mass spectrometry parameters and post-translational modifications to be considered, and then he will fire up the search. It's pretty much fast search compared to any other tool. This is the search result. So we're characterizing a 5-protein complex, and we can pinpoint where the interactions are happening. No tool has this. No tool has an interface with this. And I can click on the interaction, and I will be able to open the mass spectrum that is representing that interaction. And this is very, very important for the mass spectrometer, for the mass spectrometrist. He's able to confirm all the sequencing. He also has access to tables here showing where the theoretical ions are matching with the experimental ions. There's a degree of confidence. He can donate spectra to a global database where we have machine learning approaches to perfect our scores and make the future versions of our tool even more sensitive. So here I think now we're looking at an intralink. We have intralinks and interlinks. We also have 2D interaction heat maps, so if you want to compare two proteins. So here it's saying, which amino acids exactly did the cross-linker match. And basically that's it for 5 minutes. And this publication is coming out in a special edition for computational proteomics in the Journal of Proteomics, of which I am one of the editors. All right. Thank you very much once again for having me.
Thank you, Paolo. Questions, remarks? Up there.
Oh, excuse me. I think I don't need a microphone. Okay.
I can hear you.
Okay, fantastic. Paulo, once you have this cross-linker, actually, it's actually mediating a nearby interaction. The actual contact point may not be that way. It has to be somewhere else. But the protein is native state before. So it's more or less like anchoring things. How do you actually detect the actual contact?
Exactly. Well, there are a few types of cross-linkers, but each one with very different properties, very specific properties. In a particular case where I showed you, we're using a DSS linker, which has a length of 10 Armstrong. So if the amino acids are within 10 Armstrong, there will be a covalent bond and it will stick and it will not let go. But there are many different types of cross-linkers that will covalently bond to different amino acids, and actually we're in the process, Fábio is in the process of synthesizing some new cross-linkers too. So we can adapt very much the type of cross-linkers for what you want.
Okay.
All right. Thanks once again.
Thank you, Paolo. And now we move on, and I welcome Frédéric Veyrier from Institut Armand Frappier in Canada. And you will talk on, the title is on the board. So please.
So first, I would like to thank you for having me presenting this today. So it was a long presentation, so I should turn it a lot. So I will try to be good in 5 minutes. So I will just present you briefly what we are doing in my lab. I just have been hired at the Institut Armand Frappier in Laval 6 months ago, and this is what we will do in the near future. So I'm working on the understanding of bacterial symbiote evolution, and I will tell you a little bit later why I use this title. Yeah. Okay, so I just want to mention briefly two definitions, but evolution, I think I will pass this one. But I just want to mention for symbiosis, because in biology, this word has a different meaning than in the general language. So in biology, this term means a close and often long-term interaction between two or more different biological species, which means that there is no notion of or good or bad for one of the partner. If you want to put this notion, you can use other term. So this is why it's different when you study symbiosis when you study pathogenesis, even if the organism are the same. You have a different way of seeing things. So let me tell you a little bit what are our vision of bacterial evolution. So as you may know, bacteria can acquire new property from mutation, horizontal gene transfer, duplication, and deletion. And if this organism that can live, for example, in the environment or whatever, enter in contact with a host, and if these properties are useful for survival in this host, this property will be fixed by natural selection, and you can later on see these genetic events. And the final stage that we are looking on is the obligate symbiote. So this is bacteria that are strictly restricted to the host. So in this case, evolution has to find a way for the bacteria to transmit from one host to another. So if you look at the different evolutionary stage in relation to the symbiosis in specific genus, you can find bacteria that have different stage of evolution. Some are not infectious, and others are obligate symbiote. And if you put now the phylogenetic relationship between these species, you can see that there is species that evolve slowly, whereas others make a drop in evolution by acquiring big piece of DNA to become from non-infectious to obligate symbiote. So the question that we ask is the difference between these species, the genetic differences between these species that have different stage of evolution in terms of symbiosis, and after we detect these differences, what are the consequences of these differences? So the two models that we are using, the first one is Mycobacterium tuberculosis. So this bacteria is causing a long-term infection that can go from months to years for the life of the host. And the bacteria is living in the lung, in granuloma like that. And sometime in 1 to 10 affected person, the disease is declared, and the lung is damaged, and this causes that will help the bacteria to spread from one host to another. So here the disease is part of the process to transmit from one host to another. Now I want to present you the second model. So this bacteria is Neisseria meningitidis, and this bacteria causes short-term infection that can be for days to 2 weeks. This bacteria is living in the nasopharynx, so the bacteria is sharing this ecosystem with a lot of other bacteria. And as the infection is short, the bacteria needs to find a way to transmit from one host to another really quickly, and this is why this bacteria is taking into account the social behavior of humans to transmit from one host to another by kissing and stuff like that. But in some cases, this bacteria can go to the blood and cause sepsis and meningitis. So the difference between these two models is that Mycobacterium really need the disease to transmit, whereas here, in the case of Neisseria meningitidis, the disease is a dead end. The bacteria will not necessarily be less fit to transmit when the bacteria is causing sepsis and meningitis because it will kill the host. So it's two different philosophy of evolution. One will be quick, the other one will be slow, the disease, etc. So it's really interesting to put these two models in parallel and study them in parallel. So the tool that we are using is first we use an endless approach using like all of you new genome sequencing and specifically PacBio that I really like. And we use bioinformatic tools. Some are already there, or we also develop our own tools to answer our specific questions. And after that, once we have the differences between the species, we can take a more focused approach to really understand the role of this difference, genetical difference in the evolution of the bacteria, and use animals model after that. So I just want to mention briefly one example, a recent example, that we have done. It's a long story, so I will just spend 30 seconds on it. But what we realized for Neisseria meningitidis is that the ancestor of this bacteria undergone a cell shape change from bacilli to cocci. And using bioinformatics, we were able to screen to detect the events that were necessary for this cell shape change. And we are also studying the role of this cell shape change in the host pathogen relationship. There is some differences in the detection of the pathogen. There is also a physical property of the cell that will change also. So this is a lot of study that we have done and that we are continuing actually. No. Shit. I just want to spend a few seconds to present you my department. So I'm in the microbiology department in the S-geomorphy, and this is all the person that share my department. And we have a strong expertise in ecology, in microbial ecology, in different aspects. Some are more in the environment. Claude Gertin is working with bacteria in insect or other microbes in insect, etc., etc. And also we are really focused on with biotechnology and stuff like that, and bioinformatics too. So thank you for your attention, and if you have some questions, I will be happy to answer.
Did I understand correctly that you said that the fact that bacteria kill the host as a reduction?
In the case of Neisseria meningitidis, yes. In fact, we don't really know, but what we think is that the fact that the bacteria is evolving so quick, sometimes you have unfortunate error of evolution, if you want, and this is causing the death of the host because the bacteria is changing, is more recognized, is causing inflammation, is going to the blood. So it's just that the bacteria is evolving, and sometimes there is error of evolution. But the bacteria that kill the host, they are known to not be good to spread because.
You see, that is a paradigm that every pathogen has to be benign.
But it depends. In the case of Mycobacterium tuberculosis, it's not the case. They need the disease to.
It looks to me like it depends a little bit on the dynamics of the species, because if susceptible population is infinite, then you're actually selecting for the ones that move as fast as possible, even if they kill the host. So there is no.
Yeah, but in the case of Neisseria meningitidis, you are in competition with a lot of other bacteria for the niche and stuff like that. So this is why this example is really interesting, because it's different than Mycobacterium tuberculosis, that it's alone, or that other bacteria that are alone in their niche. Here the competition is really hard, and the bacteria has to make the place. And,
Okay, thank you very much, Frédéric. So our next speaker is Anastas Pashov from Stefan Anglov Institute, Microbiology in Bulgaria. So warm welcome. The floor is yours.
Thank you for having me. Here's the thing. Okay, so So help me, Magnus, 5 minutes. Yeah, no. The spike of HIV, as you know, is the primary target of the neutralizing antibodies. And, you know, it's a trimer, and this is GP120. And an interesting aspect of its structure is this heavy glycosylation on the outer domain. It's a cluster of 20 to 30 glycans, and it practically in the trimer it kind of fuses in kind of torus that completely protects from the outside from neutralizing antibodies. So as you know, as everything else in HIV, GP120 mutates very quickly, and so do the N-glycosylation sites, the sequons, where the glycans are attached. Obviously, they are destroyed when they mutate, but as fast as they are destroyed, they're also they re-emerge. So in the evolution, the glycan shield never doesn't have a trend down and up. It just shifts around, and this gave it the name of evolving glycan shield. Since the major pressure is from the immune system, it was interesting for us to peek behind the curtain what are the forces that move the glycan shield. To this end, we decided to study the coevolution of the positions of GP120, and these are the parameters of this multiple sequence alignment that we used. We focused on clade B. So correlation, the coevolution is measured by correlation, for instance, by mutual information. This is too easy to be useful and true. You can ask me about that later, but we are not using directly mutual information. We're using something relatively new as an algorithm, mean field direct coupling analysis, which was proposed by the group of Martin White, who was here in Cordillé. And this is the raw data kind of to the background of the structural distances in 2p4C structure of GP120, which are given in gray scale. We have overlaid the significant coevolving pairs. Obviously, along the diagonal, those positions that are close to each other or which are coming close in the tertiary structure. And this data later we presented for better analysis as a graph. Here, only the sequence related positions are outlined. And as you can see, they form a rather dense network, most of them. And especially those highly variable sequence in the variable loops of V4 and V5, in V5, and also in V1, V2, they practically form cliques. And this, to remind you, this is coevolution. It means that if one of them changes, disappears, the other one has to react to it. The V3 loop is separate. It's not so related to the rest. And then when we look at all the positions that are immediately related to the sequence-related positions, they cover 60% of the structure. So while this connectivity we see as a sign of immune pressure, you can ask me about that. This, on the other side, shows the structural relation. Obviously, the structural changes in the protein impose some kind of limitation on the glycan shield, and it has to react, and vice versa. When antibodies pressure it to change, this pressure actually is passed down to the whole structure. It has to adapt. And finally, this is another. I couldn't resist the temptation to determine the most central positions in this graph, and this is the between centrality. It's especially the top between centrality positions. Very logically, they're in the interface between the inner and the outer domain, and in the CD4 binding site actually. And the other thing, relative to our interest, the sequence, no, there are no sequence there, sequence, and obviously sequence are not related to this, how to put it, central place for allosteric interactions. It's interesting that most probably the high between the centrality positions are positions through which many allosteric paths pass. So that was all. Just this. This thing was started with Thomas Kibber-Evans in UMS Little Rock, and also with the help of Sveta Garimala, but was finished in the next 4 years in Sofia.
Thank you, Anastas. So, questions, remarks?
Yes, yes. You. Huh?
Thanks for your talk. Did you consider, did your effort consider the phylogenies sequences?
Yes.
Underlying the sequences?
Yes. It is known that this type of analysis is very sensitive to deep phylogenetic trees. So if you have kind of two branches and they're slowly branching further, this gives a false relationship between different positions. So this study is facilitated by the rich data we have on HIV. So one can prune the sequences to match best the algorithm. So here we take only one sequence per patient to reduce the similarity, those that are quasi-species which occurred in the patient. We focus on one clade also. In a way, we kind of prune this tree from both sides, so they are very, or we hope, equally related to each other species. What else? The highly homologous sequences are eliminated. So there's more than 80% homology. We have 80, no, results. But the homologous sequences are removed. And also not all positions can be studied. So actually this multiple sequence alignment was quite carefully cleaned to fit better the algorithm.
Am I interpreting it right that this type of phylogeny tree method is more robust in a situation where there are some sampling gaps or there are some lack of sampling populations, you create those big phylogeny lineage each lady positive coding sign.
No, I don't understand very well this, but the point is that you shouldn't have big groups in the sequences that are highly related between themselves. That's important. Yeah.
Thanks again. Thank you very much. So it's my pleasure to introduce Paolo Zanotto, our next speaker, coming from the University of São Paulo. So please, floor is yours. Thank you.
Thank you very much. So let's move on. This is Guarujá, is the city. Do you have a laser point? Okay. This is Guarujá, is a city near São Paulo. We're going to be studying the dengue outbreak from 2013. This is basically the dengue virus, just an overview of the cycle. It has a zoonotic component, then it goes into humans, and then it circulates among humans by means of vectors. This is the genome of the virus, 10,000 base pairs more or less. This is the E gene that we're going to be looking for because it can be very useful to show changes during an epidemic cycle. This is the state of dengue in Brazil, in the state of São Paulo. You see this kind of oscillation thing. You have cycles in where you have large numbers and slow numbers, but if you fit a very simple model here, you can see that this thing is going berserk. We're probably going to be reaching the status of Southeast Asia very soon. So it's a massive problem in Brazil. This is an outline of the study. This amount of people that were sampled. We actually got, I'm not going to go into details here because I don't have time, but in the end of the day, you had almost 1% of the population sample. This is more or less our sample density here. Here's Guarujá, it's the beach, very nice. Everybody from the state of São Paulo comes here and take dengue back to their cities. And I would like you to pay attention to this area as well, where Neymar, Pelé, they have their houses, okay? And you see that almost no cases here, so it's very interesting. And this is basically the status. Now I'm going to show you very fast I think if I click here, I should see, next one. If I click, yeah, you're going to see the, whoops. Okay. The beginning of the red and going according to time, and then it's going to decay later on. This is more or less our samples across time during the 7 months that the epidemics took place. Okay, but those are basically the cases. But then from those sampled cases that geolocated, we are able to actually derive some epidemiological parameters over time, which is very important. This is genealogy of all the almost 300 virus samples we sequenced, and those are intervention things. That this is when it was declared an epidemic situation, more or less after we reached the maximum transmission already, or reaching exhaust. This is the number of people recovered. So those are very important first order central tendencies of a SIR model for that and the behavioral parameters. And this is more or less what we infer from the genealogy. So it's very important to notice that once we have some immunochemistry data on patients that are becoming positive because of NS1 detection that's related to the amount of viremia, people in the government, they start to interfere. So this is the beginning of the interaction that follows more or less the highway system in the island. But as they interfere, the pattern, not only the rates of movement decrease, but the main hubs are deleted. This is a way of showing all those cases in terms of secondary cases. And we see basically the structure of two basic networks and several small introductions taking place. And those are the cases that we're not showing the whole shebang here, but showing case in terms of secondary transmission. You can see, so the main effect of this shantytown population here in transmission of the virus was actually the area where the intervention was done based on NS1 information. So it caused what we believe a collapse of the epidemics. So I'd like to this work was done by Christian Julian, who is in France, now working in Montpellier. He did formidable work. Jessica was working with them. And then people from Guarujá that actually helped us. And this was financed by FAPET, this project. Thank you very much.
Questions? Remarks? Is it only the DEN4 that circulates?
Yes. There was in the beginning you had the 4 serotypes circulating, but then DENG4 took over. We published a paper before we concluded this study showing the 4 serotypes there, but DENG4, the population is naive to DENG4. It took over and spread like that.
And do you have any idea why Neymar is protected?
Yes, the neighborhood where he lives, they have a private system for mosquito control. So it's a very interesting situation because around that area, we had a lot of dengue cases, and people go there to work as well. So it's very interesting that the vector control appears to be extremely important. We have the evidence for that control case.
I have a very naive question. It's what is the susceptibility of the mosquitoes for dengue4? Is it strong, better than for the other genotypes? Or?
Well, we have no idea of the competence of that. Nothing was tested, but we believe it's going very well because it's spreading like wildfire in Brazil. It caused a massive outbreak. We're just following the island, but across Santos we have as well. We have São Paulo. Today you have still dengue4 circulating the urban area of São Paulo. I didn't show data for that, but and it's changing very fast and adapting. We're following some mutations in the virus. We are collecting patients that we have dengue4 plus other virus inside them. It's messy. It's completely.
Have you isolated the virus from the mosquitoes?
Excuse me?
Have you isolated the virus from the mosquitoes?
No, we are only working with patients at this point.
Thanks a lot, Paolo. So our next speaker is Hugo Naya from Montevideo. So the floor is yours, Hugo.
Thank you. Okay, I will try to present a work that we worked together with people also from Fiocruz in Curitiba. Basically, it's the reuse of some data, and this data was a previous experiment in adipogenesis. Essentially, we compared induced and controlled samples in polysomal and total RNA, and our question was: could the alternative polyadenylation be playing a role in adipogenesis? And other question in this regard are there any difference in the 3' UTR length between the induced and controlled samples? Is the messenger RNA a good predictor of the change in proteins? Are microRNAs involved in the alternative polyadenylation? And if yes, can we develop a reasonable model for it? And how some of the model prediction relate to human variability? And in this regard, for the first question, the answer is yes. We observed a consistent and statistically significant difference in the length in the 3' UTRs in the transcript in the induced that are more longer than control samples. In regard to our second question, if RNA is a good predictor of the change in proteins, you can observe here for polysomal and total fractions that the dispersion is essentially very, very high. So RNA alone is not a very good predictor. But also we observed that the slope of this regression is far below 1. That is the number that we expected at first. But experimental evidence suggests that the correlation of the log protein is with log messenger RNA, and the coefficient of this linear regression is the translational efficiency. And if you do the math that usually we do when compared to samples, essentially the log fold in proteins, we can see that the log fold change in proteins is essentially the log fold change in messenger RNA multiplied by a coefficient that is the same coefficient that here is the translational efficient and is below 1, of course. But this not explain the high variability in the points. Then we move to the alternative transcript. We have, for example, here a gene X that has 4 different transcript isoforms, each one used in different proportion in induced and controlled samples. And if you do the difference between this quantity, you have this number here. And here we have the presence-absent matrix of the different microRNA targets in each of the transcript. 1 if the target is present and 0 if not. And then after that, we multiply this number by this matrix here and sum it up by gene. And then we have a set of regressors that are here that are additional. And then our model was now the log fold change in protein is explained by the log fold change in messenger mRNA plus some predictor here. And it's easy to see that if we introduce the effect of the microRNA in our model of the regression, that finally when we compare 2 different conditions, we arrive to the model that we described previously. In regard to the results, we can see that the models that are the first in each of the lines here and here normally explain no variance at all, less than 1% of the variance in the total RNA and the polysomal RNA. But when we introduce 1 or 2 different microRNAs, we can observe that the variance reach 30% or 20%. But more important, the microRNA that the model predicts that are important are biologically related to the process. We have searched the different microRNA predicted, and in a previous revision we found several of the microRNAs, and other microRNAs that were not described for adipogenesis were described for differentiation in stem cells. We have performed a bootstrap to check that our results are consistent, and essentially in all cases, the predicted microRNAs were performed better than the random sampling of the microRNAs, confirming the consistency of the approach. And finally, and very recently, we moved to try to combine the prediction from this model to the variability found in the human population, particularly the 1000 genome project, and we have found some interesting SNPs that in some genes that have the important difference in the frequency, for example, here in the European population and Asian and Afro population, and here more frequent in the Afro population in regard to the other two populations. Thank you.
Thank you, Hugo.
Time?
Good, yeah, perfect. Questions, remarks?
So you see that the microRNAs that are targeting OVA are also expressed at the same time, so you have the same microRNA expression and the messenger RNA expression.
Sorry?
So you have the computational work with finding the targets of the microRNA.
Okay.
Do you have also the expression of the microRNAs at the same time?
No.
Which will help you a lot because you will.
Yes, of course, this is the interesting thing of this approach. We haven't used any information about the microRNA, just the present absence, and this is the interesting that the prediction of the model without using the information, yes, correlate well with another biological information in papers. Of course, we are happy to see if when we have some information of the biological data, real biological data, this correlates with the coefficient of the model. That is essentially we predict.
Thanks a lot, Hugo. So our next speaker is Serge Alain Sadoumba from Cameroon. So warm welcome. The floor is yours.
Okay. Thank you very much for this session and for the opportunity to present our works. Okay, to present enteroviruses infecting non-human primates in Cameroon. Enteroviruses infecting primates in Africa have been rarely considered, despite the wide range of primate species that are found in that part of the world. Especially in Cameroon, there are chimpanzees who are our closest primate relative, and there are gorillas and about 28 species of monkeys. The aim of the study was to characterize the diversity of enteroviruses circulating in non-human primates, and we wanted to compare the diversity of primate virus with those found in human in Cameroon. To attain the objective, we used two stool collections, 99 stool samples from captive primates originating from primate keeping center in Yaoundé and neighborhood, and 516 stool samples from the southern and eastern rainforest of Cameroon. Viruses were identified by sequencing and phylogenetic analysis of the sequence of the VP1 gene. We perform phylogenetic analysis using distance and maximum likelihood methods. We also perform control PCR to confirm the primate species and in order to verify potential contamination of primate stool by human-derived biological material. As a result, our analysis showed that viruses from captive primates belong to various types well distributed in humans worldwide, including Cameroon. For illustration, Coxsackie viruses R13 from chimpanzees fell among human sequences that are in blue within the cluster C, which is found only in sub-Saharan Africa and in Madagascar. We also found that the Coxsackie virus R24 from gorilla clustered with human sequence from Cameroon. One interesting result is that we detected 2 enterovirus 61 in captive gorilla. Enterovirus 61 is an emerging pathogen in Asia responsible of large epidemics. The gorilla viruses cluster with human strain originating from Cameroon and Central African Republic, and both human and gorilla strain define a new genogroup which is called genogroup E, which is found only in Central Africa. The pathogenicity and the circulation of this genogroup in Africa is still to be determined. Concerning wild primates, we found that viruses in red belong to various types that were not known before this study. In particular, the strains from monkey belong to 2 virus types which define a candidate new species. And we also found that a chimpanzee was infected by a virus strain who clustered with the monkey strain in the AVG species. And this finding shows that chimpanzees, who are our closest relative, can be infected by highly divergent viruses. And since we are close to chimpanzee, such viruses have a potential to spread to humans without unpredictable symptomatology. I thank all the participants to this study and the funding of the study. Thank you very much.
Thank you very much. Comments, questions?
What is known with the mode of transmission that are expected for that eat some animals, for example, is there a risk that the captive animals will transmit the virus to the people around?
Yes, the transmission of these viruses is there is by the fecal oral route. When somebody ingests food that is contaminated by the stools, in fact, in captivity, animals are exposed to human viruses, and humans are also exposed to primate viruses.
Okay. Thank you very much. So our last speaker today is Judith Choi from Seoul in Korea. So please, welcome.
Thank you. And yeah, thank you again also for this opportunity. And I will try to keep it short. So first of all, I think I need to tell a little bit about our institute because it's a bit different from the other institutes, and it's positioned as a translational institute, which is in the middle of basic biological research and the drug discovery. So what we do in our institute, we do actually high content screening, and we developed a lot of different assays for different disease models to answer different questions, and we have capacity in terms of instrumentation and human expertise to run pretty large-scale screenings up to tens of thousands of compounds, and also we can do genome-wide screenings in siRNA. So, and because the screenings, they were done over the last at least 9 years, we have a lot of data generated at our institutes. They are mostly images obtained from the high-content screens. And what we want to do, this is the plan of the project that was initiated recently after the group for computational biology has been formed at our institute. So what one of the main goals is to reinforce what we have at the institute and try to play on our diversity, because at the institute we have biology, we have computer science, we have chemistry, and we have cheminformatics group, which is pretty kind of interesting combination for Korea at least. And so that we have different types of data, and we want to try to analyze this data in a better way, so to try to play on this diversity and to create a more context-rich database, which has this kind of name, which is Discovery Explorer database, to support the screening and biological research at our institute, and also to produce maybe more interesting things. So the main point here is that we will try to analyze images in such a way that we will extract information about the cellular morphology, because this is a cellular assay, and in one plate you might have actually millions of cells, and because every cell is different and its response is different to the same treatment, we need to have a good way to actually quantify this response in order to first of all understand what are the normal cells and what are the abnormal cells, because there are also some rare effects that in the well that we have in high-content screen, and we need to be able to detect those events. And on the top of that, we also have quite lots of noise in the detections. So in overall, the project is divided in 3 phases, and currently we are in the first phase, and we are currently working on making the prototype of the method, first of all, actually to check if it works or not, and to identify the most weak points that we have. The idea that we also had in mind when we started this project is to have something that is based on the incremental learning, something that is not fixed from the very beginning, but that can actually evolve as more data arrives. And at the end, if everything goes well, there are actually a lot of different ifs. If everything goes well, then based on the morphological response and the different types of morphology that we can extract from the screen, we can actually try to annotate compounds. And we actually can try to build the predictive models between the compound and the morphological response. So that this project, in fact, it is very challenging in terms of what we want to achieve and in terms of the time that we have for this project. But if it succeeds, it's going to be very interesting. Well, in any way, if it's successful or if it's not successful, it's a very interesting thing to work with. And in the worst case, we will have improved the infrastructure at our institute, much better infrastructure than we have, which is actually not that bad worst case. But in a good case, we will have actually like really interesting tool that might be working. So that's yeah, well, I think I should end here because I can't run out of words. Yeah, okay, thank you very much.
Thank you, Judy. Comments, questions?
A question concerning the high-content screening cells. What kind of cells would it be? Different types of cells?
Well,
because there are some populations where you will have different subpopulations, so they will definitely behave completely differently. So if you are going to, within this idea that all these people that defend the idea that the single cell screening or single cell analysis are a good thing to go through, then how would you cope with that different populations that are in subpopulations that are in there? Meaning that you will how enough would be enough to see.
Okay, yeah, that's a very good question, because there are different cell lines, and basically they respond differently for the same treatment, and of course they might have very different subpopulations. So that was the idea for the morphological learning, so that we will try to identify the clusters, basically that represent these subpopulations. We will try to learn these clusters automatically, so that when we have the image, we can actually profile it and say how many, what is the proportion of cells that belong to this cluster or to that cluster, etc. So that basically the subpopulations, we will try to represent them in our models. And for the pilot version, we are working with one fixed cell line, with one fixed assay, to ensure that at least the initial conditions, we can assume that they are the same. Well, otherwise we would just, I think it would be impossible to make different cell lines. But if it works, actually it can provide some very interesting things and very interesting insights. For example, if you have two different cell populations, cell lines, and then we have the same compound that we use to treat these cell lines, then we can actually check if the morphological response is the same or not in terms of the subpopulations, cell shapes, and some other things. Yeah.
And just, sorry, technical complementary question. Will there be, because I've worked with images with cell lines, and we had the opportunity to have a whole apparatus, I would say, that is related to a temperature control. So the cells were on a 37-degree chamber, if you wish, or 34, depending on the cell. So are you providing this kind of, or are you planning to include this kind of temperature control for each cell line? Because otherwise you would have different shapes of these cells,
yeah.
even if when 1 degree of difference.
Of course.
And then you change.
Of course, yes. Well, basically we are not developing the assays. We are working with the assays that were developed by biologists at our institute. And as assay, well, there is SOP, which actually strictly fixes everything, so that we assume that there were some specific temperature conditions, and we will have access to the SOP file. So that, for instance, if biologists want to take a look at what is this cell line, what are the conditions, and how it was incubated, yeah, we'll work with this. Thanks.
Okay, Jury, thank you very much. I want to close this session by thanking, first of all, the people that have been instrumental for putting it in place. So Tony Borderia, Willy Bordeaux, Julie Lay, Jacob, Gabriel, and Rasmus for helping out here, and the entire IGDA team in Institut Pasteur in Paris. And I also want to thank the audience, all of you, for coming here, and the International Network for Data Analysis at Institut Pasteur for collaborating, and I truly look forward to our collaborations in the future. I think we have a great opportunity, and I think we had a really good start. So thanks to all of you. Thank you. Now there is a cocktail outside, same place as the coffee, so please take an apéro before you go for dinner, right? See you outside.